Okay, so today we're going to talk about cancer, and I'm going to be very honest. There is so much to learn about cancer that what I will be teaching you will just be the basics. We're going to learn about stem cells, and you may have heard about those in your regular bio class. We're going to talk about regulatory genes, which are genes you have in your body or hopefully have in your body that will regulate the control and spread of cancer. And then we're going to also talk about treatments. But before we go any further into cancer, we're going to look a little bit closer at some chromosomes and talk about some pieces and parts of chromosomes that play a role in cancer. Okay, so a regular chromosome, you've already learned, is made of a single long thread of DNA, okay? And then if a cell is going to divide, it's replicated and made an exact copy of itself, and that is a sister chromatid and a sister chromatid. They stick together, and then they go through the division process together, okay? And all that happens during the M phase, right? And if you're confused about that, go back to the video about mitosis, and it'll explain all that's going on. The one thing I did not address during mitosis video is that at the end of each strand of DNA, there is DNA that actually does not code for anything. Okay, so no genes, does not code for any genes. Okay, so the blue is where your genes are, and the red is still attached, is still one long piece, actually has no coding there. These are like buffers or protection, so that as your chromosome is moving around and dividing, if the tip ends get damaged, oh well, okay? Because this information really doesn't change anything. So that's why all of your textbooks have a nice hard cover on them, because it's going to protect the inner pages and make sure that the inner pages don't get damaged. Now, when you're born, your telomeres, okay, so that's what these are called. These are called telomeres, are as long as they'll ever be, okay? And so they are there to protect the ends of your chromosomes. And the theory is that the length of the chromosome is basically your life expectancy. So if you're born with little short, tiny telomeres, then it won't take very many divisions before your actual DNA that is necessary starts to be exposed. Now, it doesn't mean it'll get damaged, but it definitely means that right about here, this is when, the good stuff gets harmed. And this is where older people who have aged, you start seeing more and more and more issues with them. Younger kids, down here, your telomeres are protecting this DNA, hopefully. And so it keeps these chromosomes intact. Now, when we're looking at chromosomes, I can see your whole chromosome, but I can't tell the difference between what's a telomere and what's a regular chromosome with my naked eye. If you were to send your DNA off, there are people who say that they can tell you how long your telomeres are. Now, that's assuming, you don't have some sort of crazy accident happen. That's just like how old you would get. And different cultures are known to have different lengths of telomeres. And so if you come from a family that lives a long time, you probably have long telomeres. Okay, so when your cells are going through division and they divide, and I'm telling you, like a little tiny bit just gets removed all the time. This also limits the max number of divisions, okay? So this cell right here, when it goes through mitosis, it divides into two new cells, okay? And the theory is, and the math works out, The limit is called the Hayflick limit. Okay, and that cell and its lineage can maximum divide, this is kind of a big range, but between 40 and 60 times is the max amount of times, and that's estimating how long it takes to degrade this whole telomere. All right, so if they're short, it only gets about 40 times before the cell then gets to the blue part here and it can't work anymore. So the lineage of cell here, maximum 40 to 60 times, and then after that the cell dies. Now, that's aging, okay? And so, we just don't know how long your telomeres are. Now, I've been asked, and every time I teach this topic, kids are like, well, can you add to your telomeres? Would that add to your life? The answer is yes and no. When you were created, okay, you were created from an egg cell and a sperm cell. Okay, so egg cells are created when mom is literally being developed inside of her mom. So those egg cells are pretty new. Sperm cells are produced every day. They take about 30 days to be created, and they are the age of the father. So the chromosomes could actually be shorter than normal. But inside this gamete production, there's an enzyme, and the enzyme is called telomerase. And what telomerase is going to do is it's going to add telomeres to these chromosomes in the sperm cell and the egg cell, so that any new offspring that gets made is going to have long telomeres. Because you want your babies, oops, not an S there. You want your babies to have the longest telomeres as possible so that their cells can divide the maximum number of times. All right, so the enzyme exists, okay? It's there. So it's possible that we could potentially add telomeres to our chromosomes. The problem is you have billions of cells in your body, and you would have to do it to a lot of cells. So the science would be more appropriate that you take an egg cell or a sperm cell, you measure and you add the telomeres to those cells, and then you get this cell that has like a combination of extra long telomeres. Now, definitely have not done human testing on that, but I don't know the science behind who has been studying telomerase, but I wouldn't be surprised if there was some mammal research where they were seeing if they could increase the life expectancy of some other mammals by adding this telomerase. Now, the catch on this is if you go back, flip over here to our Hayflick limit, this is important because tumors and cancer, which is what this video is about, are literally just huge masses of cells that have divided and divided and divided and divided and divided. So wouldn't it make sense that after 40 or 60 divisions, the cancer cells should die? Well, that's because cancer cells make their own telomerase. Okay, so they can actually add to their chromosomes so that they never have to stop dividing. And so potentially the answer to aging could be in these cancer cells. So we're now going to spend a little bit of time on talking about what happens with cancer cells and how it happens and how we treat it and those kind of things. Now, with notes, I've had some people say, you got all kind of stuff going on. You can take notes however you want to. You can have one page where you draw a lot of things, and then you can have another page where you just write notes and take bullet points. One of the things you're going to learn is sometimes people give you PowerPoints and everything's nice and written out for you, but it's really good to learn how to follow somebody, pause the video, write things down, pause the video and write your questions down so that when we meet, or you could send me a message and say, hey, what does this mean? These videos are for you so that you can slowly take in all the information and kind of let it digest and marinate and think about it. And then it's much better when we're in person and you can ask questions in real time. But this is also a good format because you get the whole thing without any crazy interruptions. Okay, so I'll do the correct and then the incorrect, because cancer is obviously the incorrect way of things occurring. Okay, so going back to this picture of an egg cell and a sperm cell coming together, and it creates that first diploid cell. Okay, so I'll go back through that vocabulary real quick. In humans, egg is 23 chromosomes and the sperm is 23 chromosomes. And so they call this haploid, and they call this one haploid because it has half of the DNA. And then when they come together, you get the 23 plus the 23, and you get the 46, okay? And this is the first diploid cell that exists as you, all right? So that is going to be, and it divides and divides and divides. And I told you in a previous video, okay, so this will divide into 2, and then 2 into 4, and then 4 into 8. Okay, and then you become just a nice big ball of cells. And then each one of these cells will differentiate. And prior to differentiation, these are all called stem cells. Now, stem cells are all your normal body cells. They just not have specialized yet. So in order to specialize, they're going to have to regulate which genes are on and which genes are off. Okay, so let me flip the page here. So the process is very complex, but if you were to look at a chromosome in any of your cells, segments of that DNA are called genes, and not all cells need all the same genes. So you have approximately 25,000 genes in your genome, okay, in your genome. Okay, and your genome is your full set of chromosomes, and every cell has the entire genome. This is crazy because you've got every single recipe you need in every single cell. So it would be completely wasteful to have all of that accessed. So what cells do is they turn off unneeded sorry, genes. And the way they turn them off, they don't have light switches, so they methylate them. Okay, and methylate means to add methyl groups. Okay, and so literally, like right here, if this is a cell and I want to keep gene number 1 here on, but this segment is gene number 2, and I want to turn gene number 2 off, they trigger a signal. So there's a signal transduction pathway, and what happens is some methyl groups are going to be red. They attach these methyl groups to that segment of DNA. And so what it does is it's like if you had a textbook and I said, you don't really need to do chapter 7, okay? Instead of tearing out the pages, like some people, you would take that chapter 7 or whatever, that section of your book, and you would glue all the papers together, okay? So it's still there. And if you were very patient, perhaps you could unglue each sheet. But the methylation cuts that gene off, and it makes it inaccessible, and therefore it doesn't clutter up the gene. So now it's like it's all locked up and closed up. You don't have to worry about it. So cells would turn genes on and off, and then when they do that, they become specialized. Okay, and that process of doing this, of turning these genes on and off, is called differentiation. They become different. Okay, so that big ball of cells that was you, each one of those cells has to specialize at some point and become different so that it can do its job. And maybe this gene is something that the liver cell needs. So the liver cell would keep this one unmethylated, but this I don't know, skin cell locks it up because it's not necessary. So by methylating the unnecessary genes, the cell becomes specialized and it cannot go backwards. Okay. In life, when the stem cell specializes, okay, and it differentiates, it doesn't go backwards. So it's decided. Okay. Thank goodness we don't have that. In first grade, you get to choose what you're going to be when you grow up, and then you have to stay that. Okay. That's kind of what this is like. We get to change our mind. So in your body, you have these stem cells. And I told you that your brain has a nice little package of stem cells dead in the center that have not specialized, they haven't differentiated, and so they can actually turn into new brain cells. And if we access them, I wonder if we could take them, just like we have donors that donate lungs and heart and skin and eyes and things like that. I wonder if we could donate these stem cells and give them to someone else who's had some sort of problem or whatever, and see if we could get their stem cells to differentiate for them. Okay, not sure. Now, in your body, nice little gingerbread man here. Okay, in your bones have a lot of stem cells too, because your bones are constantly making all your blood cells. So inside there, they make red blood cells, white blood cells, they make your platelets, they make some of your immune cells. So inside the bone cells, there are stem cells, and they're called pluripotent. Okay, pluri means they can make a couple of different types of cells, but they have partially specialized. Okay, so the bone marrow cells only can make like 4 or 5 other types of cells. So they're pluripotent. They plural. They can make a few things, but they could never turn into a heart or a liver or a kidney or any other cell because they've partially specialized to this point. In your brain, these cells are, I think they might be totipotent. I'll have to check that one, okay? But totipotent is what we start out with as a ball of cells, and these can become anything. And the root word here is toti for total. So totipotent stem cells are the cells that actually can become any cell in your whole body. So when you started out as that ball of cells, that hollow ball called a blastula, those were all totipotent. All right. In your skin, where your hair and skin cells grow, there's some pluripotent stem cells there because they've partially specialized and they can only make like certain things. You have some stem cells in your heart that are reserved for repair of just your heart. And I would dare say that scientists are probably going to find that there are lots of locations of stem cells in our body that are just kind of hanging out there waiting to do repair or replace, so that the ones that are actually working never actually have to take a break and do mitosis. Because the deal is when they take their mitosis break, then they're not working. Stem cells, okay, they are the reserves in your body as an adult, okay? So they are not working at all. So those stem cells that are deep in your brain, when they divide, we aren't losing any brain activity or productivity because those cells weren't working anyway. They were just standing in reserve, waiting on those sidelines to see if they were needed. Okay, so with all your cells, the goal is that they follow directions and they do their job and they stay in their place, and all these cells work perfectly. And some of you, that's going to be true. You'll never have cancer. And we're going to talk about what cancer is, how it occurs, why it occurs, and why it's so difficult to treat and monitor. Okay, so there are some rules that all cells follow except cancer cells. So cancer cells are the ones that just don't listen, okay? So here are the cell rules, okay? Number one, they follow this rule and it's called density dependent limiting or inhibition. I'll call it inhibition. Okay, so normal healthy cells that are not cancerous, density dependent, that means crowded. They will, inhibition means like stop. So normal cells will stop dividing when it's too crowded. They send signals out to each other in the vicinity and they let each other know like this is way too crowded, like something's up, like stop dividing. Okay? Cancer cells do not follow this. Okay, so cancer just does its own thing. Like they love crowds, okay? And that's why they can actually form tumors because a normal cell will not get all crowded. But cancer cells are like, whatever, we don't care. We're gonna form tumors, okay? So rule number one, they don't listen there. Second thing is there's something called anchorage dependence. Okay, and anchorage means to be attached to something. So most cells, most cells, because some of your red blood cells and some other ones are free flowing, but most cells have to be anchored in order to live. So most cells will not divide if not anchored in the body somewhere. Okay, of course, cancer doesn't listen to that. They can flow, they can move, they can do whatever. And so they'll actually divide in the bloodstream and do all kind of things. So once again, these two rules are in place that all your normal healthy cells will follow. But cancer loses the ability to pay attention to these things. And we'll talk about why that is. And it has something to do with the damage that occurs. All right, so normal healthy cells, how do they become cancerous? Okay, we have these things. Carcinogens, okay? Sounds like a scary word, and it literally probably could be just about anything in this world. But carcinogens is a term used for anything that causes cancer. And in the short little life that I've lived, it's been crazy, all the different things that they have told us that, oh yeah, sorry, now those might be cancer causing. A big one that they're pointing to now that, if it's true, we're all done for because plastic, they're saying plastic bottles, plastic containers, heating things up in plastic might have some sort of carcinogens. What I'm here to tell you is that some of these basic life exposed things that we call carcinogens, you actually might have a natural protection to. Now, when you do these extreme things with, smoking and literally smoking anything, it doesn't matter what it is, you're putting hot burning chemicals into your lungs. So whatever it is that you're doing with inhaling smoke is damaging your lungs and killing lung cells. That's going to cause some DNA damage. Okay. UV radiation. If you're out in the sun a lot and you're not wearing sunscreen and you're not blocking that ultraviolet. Now, regular white light is fine. That's what causes your skin to tan. Tanning is a response to the sun and it's a response to the white light. UV radiation is the light that damages and destroys your DNA. And when your DNA cells get destroyed, that's where you get skin cancer. Skin cancer can actually begin, cancer can actually begin at the skin level, but then it might travel and go somewhere else. So you might have cancer somewhere else that actually came from this UV damage somewhere. Same thing like with gel nails. Boys, y'all probably don't know what that is, but girls, you go get your nails done and you put your hand under those UV lights. That is some UV damage. Now, a little exposure should probably be fine, but you never know, right? X-rays, when you go get an X-ray, they tend to only get an X-ray of the image in the area that they need, and they'll lay a heavy metal vest over the rest of your body, and that is to block all the other X-rays because X-rays, excessive use of X-rays have been known to cause DNA damage. All right, and then we have viruses. All right, viruses, their whole job is to get inside your cell and insert their DNA into your cell. So by doing that, they might actually damage your DNA. And we know specific viruses that actually are cancer causing. And one of them is the HPV virus, the human papilloma virus. And most of you have, that might be an S, that you might have the vaccine called Gardasil. And Gardasil is a shot you get, you think you have to get 3 of them. And what it does is it blocks this virus from getting into your body, which blocks this DNA damage. Okay, so anytime someone refers to something as a carcinogen, it just means that it is potentially a cancer-causing agent. And your main ones are going to be your UV radiation, smoking, alcohol, and X-rays. Viruses is definitely right in there, and there's probably a lot more damage that viruses do than we realize. And that's one of the concerns that people have about coronavirus is what are the long-term effects? What changes is it making? And so, when that vaccine comes out, that's definitely something we want to look into, because if we can block those viruses from getting there, then we block that possibility of cancer. Now, to backtrack real quick, this HPV vaccine for Gardasil, this is a sexually transmitted disease, and in females, it tends to cause cervical cancer. So if you can get that vaccine, it keeps you from having cervical cancer. And cervical cancer can lead to cancer in your uterus and your ovaries and all the different reproductive organs. Okay, so let's talk a little bit about cancer. We already know these cells are your cells. And that's the crazy thing, is that cancer cells are literally your cells. All right, so if there's lung cancer, these are lung cancer cells. So it's not like it's some crazy, weird pathogen that has come from the environment in your body. It's like, oh, look, there's something that shouldn't be there. Cancer cells are yours, okay? And they have all of those markers on their cell surface, those carbohydrates that tell you that they're friendly. So normally, this can be kind of a problem if they're really good at hiding. I will tell you that cancer cells, because they have some sort of damage to them, sometimes their membrane proteins don't match. And that's good, because if their membrane proteins don't match, then your immune system will pick up on it, and your immune system will tag it or target it or tell it to kill itself. And the process of a cell killing itself is called apoptosis. So right now in your body, you do have cells that have gone rogue. They've been damaged. They're dividing way more than they should. Your immune system picks up on it. Okay, and your immune cells are always flowing through your body over and over and over and over. And they recognize that this cell is not what it should do, and it tells it to kill itself. But there's other things that are going on that that can help you as well. And it's all about the way the cancer cells behave. Okay, so I'm going to add a little list here for you of all the bad things about cancer cells. Okay, so cancer cells. All right, number one, they don't follow the rules. Okay, so cell rules. All right, and so the chemical that is required to get a cell to divide, there has to be some sort of signal, and those are called cyclins, and they're ligands that trigger division. Well, if a cell is dividing uncontrollably, it's probably not getting a regular normal signal. It's probably getting a signal from somewhere that it shouldn't. And the crazy thing is that cancer cells can make their own cyclins. So not only can they signal themselves, so they can do autocrine. Okay, so here they are, and they got the little membrane receptors, and they secrete the signal, and then they signal themselves to divide. They can also do paracrine signaling, which means, they're secreting this chemical, so any cell in the vicinity that also has those receptors is also going to respond. So they can create this whole chain reaction of division of cells. This was a good guy, but all he's doing is just responding to these cyclins that this crazy damaged cancer cell is releasing. He's not being bad. He's responding to the signals that are being sent. Okay, so then, but your cell has these genes, and they're called regulatory genes. And specifically, we have some called tumor suppressor genes. All right, and they recognize when this stuff is happening. And our most common tumor suppressor genes is a gene called a P53, and then there's a gene called a BRCA gene, and then there's one more. There's a RAS gene. Okay, so these three you need to know. Okay, there's a BRCA1 and there's a BRCA2. Okay, and these genes are in your DNA, and they do what it sounds like: tumor suppressor. When your body recognizes that a cell has become cancerous, they turn these genes on. And P53 genes, they trigger a protein, and this protein inhibits the cell cycle. So it inhibits mitosis. All right, so when P53 gets called to action, he's going to inhibit mitosis in that cell. We want a lot of P53 genes, and we want good P53 genes. What they have found is that some people are born with a mutated P53 gene, and if your P53 gene is mutated, that's basically like nobody's home paying attention to what your cells are doing. This gene is probably is responsible for a mutated P53 right now we know is responsible for more than 50% of all cancer. Okay, so your body should be able to recognize, but because your P53 is mutated, then it doesn't recognize, and then it doesn't inhibit mitosis, and then the tumors start forming, and you have all kind of issues. So your P53 gene is actually referred to as the guardian of your genome. And that's kind of a cool thing, because this is like your guardian angel gene. All right, so that guardian angel gene is constantly being turned on and off depending on what's going on in your body, and making sure that it stops and it inhibits that mitosis. Another quick thing that P53 can do is it can activate suicide genes. So he's like the guardian of the galaxy here. Like, he can make a lot of things happen. So he can inhibit mitosis, or he can activate suicide genes. He can activate a gene called P21, and P21 repairs DNA in cells or in cancer cells. So he not only halts the mitosis, he triggers P21 to come in and repair the damage, so that not only are we making sure it doesn't keep dividing, but then if it happened to divide again or something else, the DNA is now repaired and it's not cancerous anymore. So repairing the DNA, halting or arresting mitosis, and triggering apoptosis are the three things that P53 does. And remember that and they have found that some animals have extra P53 genes. So if you have extra P53 genes, then you're going to actually be so very good at suppressing any type of cancer in your body. So if a human had extra P53s, then they're going to just be better at it. And these families, these genes get passed along. And so if you don't have a lot of cancer in your family, you probably have some really good P53 genes. If you're seeing a lot of cancer and a lot of damage at early ages in your family, they're going to be maybe an issue with your P53. And so you want to be super careful about all your exposure to these different carcinogens. Okay, another thing about cancer cells is that number 3, they do not listen to checkpoints. Okay, so in the cell cycle, in that video, we learned that there's a lot of checkpoints along the way that keep a cell from moving to the next stage. But cancer cells just don't listen to them. And it's probably because they've removed their receptors. And so if you don't have a receptor, you literally cannot receive the signal, and therefore you just move straight on through without paying attention. So they don't follow any rules. They're rule breakers. Somehow they're able to evade apoptosis if you let it occur and happen for too long. They can actually avoid killing themselves because they'll also remove those receptors. And at this point, I always compare it to teenage kids. Your cell phone, if you do not want your mom or your dad to get in touch with you, you turn your phone off. And then all of a sudden, like, you know it's coming. You know you're going to be in trouble when you get home. But for that time being, they don't know where you are and they can't get in touch with you. So you have literally removed those receptors, because your cell phone will be your receptor where you would receive that signal from your parent, and you are evading those signals. And so at some point it does catch up with you, but for a little while, you're not receiving the signal. So you're not going to respond. And if you didn't know, how can you get in trouble? Remember, you're always going to get in trouble. Okay, so, and here is just a crazy thing. These cancer cells, we're not following rules. We can make our own signals to cause ourselves to divide. We don't listen to checkpoints. We evade apoptosis. These are just bad dudes, okay? They just don't listen. All right, and the number 5 is they can signal blood vessels to reroute and directly bring them nutrients. Okay, so cells that are dividing require a lot of energy, and if they have to wait on the regular supply, that's not very fair. So what they do is like, you know what? Let me just get my own blood supply straight to me. I can get the nutrients first, and then I can use them to do what I want to do. So this is gonna take all your regular nutrients, and this is where one of the first signs of cancer is people start losing a lot of weight, like for not even trying. Like they just start losing a lot of weight, and it's because these tumors have rerouted the sugar to them, and they're burning up all those calories in the division process, because cell division is very expensive calorie wise, and so they use up all those calories, burn them up. You start losing weight because the calories that you were eating are not actually going to your body. All right, so this is cancer, and this is the biggest problem, is because it starts off as your cells. It's in your body, and if you haven't learned anything yet, what you should learn is your cells are unique to your body. So cancer treatments almost necessarily need to be specific to the patient. So one person who has breast cancer and another person who has breast cancer, yes, you both have breast cancer, but no, you don't have the same cell membranes. And so treatment for one person is very successful, and treatment for another person might be moderately successful. And we're going to talk about why in just a second. So these are going to be, how do we stop cancer? And every year the kids are like, I think the government has a cure for cancer and they just don't tell you because they wouldn't make that much money. It's not like that. We know what causes cancer. We know how to fix it. We don't know your cells And so we have to specifically target your cells. And the bad thing is that cancer takes a big hold of your body a lot faster than before you even realize it's there. So if you can recognize it early, then you can get rid of it, and it's not nearly as big of a deal. But if cancer is allowed to, especially lung cancer starts so small, and lung cancer, it has a awesome capability of flowing and moving, and so it spreads rapidly. And that's why I'm so against you putting these things in your lungs and creating that problem from the beginning. Okay, but it's there. It's your cells and it's your body. So when scientists do recognize it, they have to make it very specific to you. All right, so scientists have pretty much stated that in order for a cell to become cancerous, it's literally an accumulation of mutations over life. Okay, and so we're talking about cancers of old age, and the estimated is about 6 mutations must occur before a cell becomes cancerous. Okay, and the mutation usually occurs in, we call them proto-oncogenes. Okay, proto means good. Okay, so oncogenes are genes that should be an O. Oncogenes are genes that regulate cell division. Okay, when they're the bad guys, we call them oncogenes. When they're doing their job, we call them proto-oncogenes. So if you mutate one of these genes that maybe triggers apoptosis or is a tumor suppressor gene like your P53, okay, then you're going to have these mutations that are possibly going to cause this cancer. Now, does that mean you're always going to get cancer? No. Some of you may have a few mutations now. You may never have another mutation. You may be good to go. But if you continuously expose yourself to scenarios that can cause these mutations, then you are continuously pushing yourself closer and closer to this number. And when you hit that number, that's when those cells start changing. Now, could things happen that are out of your control and something has occurred that you're not doing anything wrong and cancer still happens? And the answer is yes. If your P53 doesn't do its job, or sometimes a virus gets in there and damages that cell and causes it to be cancerous, and so that's out of your control, and so you need to go in and have like a tumor removed or whatever. Okay, so viruses play a role in about 15% of cancer. I told you P53 is about 50% of all cancers. There's a gene called RAS. I referred to it, but I didn't really say much more about it. Your RAS gene. Okay, it stimulates the cell cycle. So if for some reason it goes rogue and it becomes hyperactive, the hyperactive RAS gene is responsible for about 30% of cancers. So everything in cancer is happening here at this minor level, tiny microscopic level. But these are your big contributors to what's going on. And the reason those happen is because of mutations, because of damage that you do to your body. All right, so real quick, you get cancer and you want to know how to treat it. There are a couple of things that we do. Number one, radiation. All right, and radiation is specific spot. Okay, so it's literally like wherever that mass of cancer cells is. So radiation is only good in tumors that are sessile. That means they're not moving. And so we radiate that one spot. It damages the cell beyond repair. Okay, so basically we're just like zapping it, trying to kill it, and then you want to try and remove it. Okay, this is tough on your body because the radiation is, we're trying to be very specific, but you can't be, you're going to kill some of the good tissue around it too. Okay, 2 is we just remove tumors, which will be, it was very good. So if you have a tumor this size, we're going to remove all of this because we want to make sure we get the bad stuff and then anything around the periphery that maybe it triggered or turned into a cancerous cell. So little tiny freckles on your body that turn real dark and start getting cancerous, you're not going to get just a little tiny freckle removed. You're going to get that whole space removed. And when they stitch it back up, it's going to be bunch of stitches right here to get you to close back up. So wear your sunscreen. You can still get tan, but block that bad UV radiation. Another thing we do is chemotherapy. Okay. And this is chemicals and it's therapy. And I'm going to tell you, this used to be the worst part of cancer. People would decide, you know what, I'd rather just die of cancer than do the chemotherapy because they would put up to 80 different chemicals into your body hoping that something would work. Because remember, all our cells are so different and they just really didn't know which chemotherapy would work for you. We have gotten so much better at that. And now there's a protein test, okay? And they test the proteins on your membrane and they can determine which chemicals would actually work for you. So it's very individualized and they can say, okay, so Ms. Frick, you will not respond to chemicals A, B, C, and D, but chemical E, you have those membranes on your protein. So that will be a really good chemotherapy to use for you because it's going to respond very well. The way they would know that is they would have to biopsy. And biopsy means to remove some tissue from the body. They would look at the proteins on the membrane and then they would determine that. The problem with biopsying and all this process is it takes a little bit of time. And the more time you take while you're sitting back waiting, cancer is still dividing and dividing and dividing and dividing and growing. So the best thing I can say to you is you want to take really good care of yourself the whole way through. You want to make sure that you're not doing damage to your body so that your cells can regulate themselves nicely and they can take care of yourself and you don't have all these things going on. Now, I'm going to tell you on page 338 of your book and 339, it shows you, so page 338 and 339, it shows you different breast cancer treatments based on the different breast cancer cells that they have recognized. And breast cancer is the leading cause of cancer in females in the United States. And your breast cells have receptors for estrogen on the surface, which is a normal hormone for women to release. Estrogen triggers cell division. And so it shows you the different types of chemotherapy that they use based on what receptors you have or don't have. And if it's a cancer cell, you know that the receptors are going to be a little bit different than they should be. All right, so that's a lot of information. I hope that it helped you, and it's probably going to trigger a lot of questions. So while you're watching this video, make sure you go through and you write down any questions you might have, and I will be more than happy to answer them. But cancer is basically your cell cycle happening way more than it should and without any regulation. Have a fabulous day.